present a paper regarding contamination of aromatic herbs and medicinal plants by fungi and associated mycotoxins. For preparing this paper, I structured my motivation on 3 pillars. One is regarding consumption dynamic. The second, diversification of commercial products, and the third one, climatic changes. All of them affect the quality and the quantity of medicinal herbs and aromatic plants. Let's see a few words about each one. Regarding consumption dynamic in European countries, we have to take into account the social changes, then the food diversity. Now we assist to a huge number of food products. Now then, the seeking for new flavors. Everybody is seeking for new taste. That means new flavors. That means using new aromatic plants. Then, increased interest in ethnic cuisine. We all travel, and when we are traveling, we want to taste special food specific for the country where we are. Then, processed foods that require special spices and herbs for cooking. Tourism is a main contributor to consumption dynamic. Then traditional restaurants of various ethnic groups. Very important are the immigrants involved in supply chain stories, because they come with own aromatic herbs, own spices from their native countries. And in fact, they are putting on the market new spices, new aromatic herbs for the places where are living now. Then catering systems, and in the end, but not the least, it is this increased interest in healthy lifestyle. That means we all want to use natural additives, natural compounds instead of synthetic ones. Of course, in this picture, very important is the media contribution, because it stimulates the demand by a great variety of culinary programs, special magazines, special organized contests, and sometimes by radio programs. Now, I want just to highlight how the producers and the consumers change a little bit at European level. Until 15, 20 years ago, the main producers of aromatic herbs were France, Italy, and Greece. And now, because it is a great interest in these aromatic plants, great producers are Germany, are Poland, are Austria, Bulgaria, Spain, of course, and Hungary. And very strange that the consumers the great consumers are United Kingdom, Germany, Romania, and Hungary. And all these 4 states, they use about 58% from aromatic plants and spices placed on European market. This data is according to Eurostat study from 2010. What I wanted to highlight that due to these factors affecting, spreading aromatic interest in aromatic plants and medicinal herbs, now countries that didn't have tradition in producing this type of plants, now they are important producers, and others, of course, are important consumers. Regarding the diversification of commercial products, we know that aromatic herbs and medicinal plants, they may be commercialized individually or packed in special bags with special mix for some dishes. For instance, for pasta, we have a mix for obtaining pasta sauce. Or we have sauce mix, or we can have food supplements. And finally, we have food products, where we use these aromatic herbs. And then, until 10, 20 years ago, aromatic plants were used mainly for meat products and for balsamic vinegar. Now, they are used in a large range of products, including cheese products, including spirits, including juices, flavored oils, bakery products, canned food, and confectionery ice cream, candy, and chocolate. Now, the third pillar was regarding climate changes, and climate changes has to be regarded all together. That means we have to take into account all the changes regarding temperature, regarding rainfalls, regarding the frequency of rainfalls, the dry years, and of course, we have to take into account that due to climate changes, we have increased pollution with carbon dioxide and nitrogen oxides. And due to that decreasing of ozone content from stratosphere, we have also increased UVB radiation. All these factors, they affect the diversity of fungi that can develop on these aromatic plants or medicinal plants. And not only the diversity, but they can also affect that relationship between the host and the pathogen. Now, we state these three important problems, and we saw that now we have aromatic plants in all the Europe, not only southern part of Europe. We have large consumption. We have different products where we use these aromatic herbs. And now let's talk about fungi contamination, because all these aromatic plants and medicinal plants, they infect our plants. That means they can be attacked by microorganisms like bacteria or fungi. And when these microorganisms develop on plants, they can synthesize some toxins, not all of them, only part of them. And in our study, we are speaking about mycotoxins, and mycotoxins are synthesized by fungi. And mycotoxigenic fungi are belonging especially to genera Aspergillus, Aspergillus, Penicillium, and Fusarium. Of course, not all the species. Now, I mentioned mycotoxins. What are mycotoxins? Mycotoxins are compounds that are synthesized by filamentous fungi. This because they are not important for fungi development. They are called secondary metabolite. Maybe they interact in the whole ecology of microorganisms, but there are not certain studies for it. What I want to mention that they have different chemical structure. That's why they have different effects. and they are small molecules. And another very important thing, they are some of them are very stable regarding temperature. That means it is not comfortable to say, okay, I have mycotoxins, but I put my product in the oven, and there I have 200 degrees, and my mycotoxin is gone. No, it is not gone. Some of them are resistant up to 250 degrees, 230, 250 degrees. And very important also is that they are active in very, very small amounts. That's why the regulation regarding maximum acceptable level, regulation of European Union, of course, for different commodities are of ppm or ppb level. That imagine how aggressive are that they are able to be active at such low concentration. To have an image, try to imagine that you have 2,000 corn stalks and there are only 5 seeds contaminated. This is ppb level and ppm level, and for ppb you go 1,000 less. It is very important to know that they are active in very, very low amounts. Who can produce mycotoxins? Now there are over 400 mycotoxins known. But not all of them are aggressive. The most aggressive microorganisms are mentioned here. There are aflatoxins, aflatoxin B1, B2, G1, G2. M1, it is not caused it is not a metabolite of fungi, filamentous fungi directly. M1 is found in milk. Usually, it is obtained by metabolization of another type of aflatoxin. And then, ochratoxin A, deoxynivalenol. Deoxynivalenol belongs to a larger group called trichotersans, and zearalenone and fumonisin. And they are producing by fungi, as I told you, belonging to Aspergillus. Aflatoxins, especially Aspergillus flavus parasiticus, ochratoxin A, especially Aspergillus ocraceus carbonarius, in wine, Aspergillus carbonarius, especially Niger, and Penicillium verrucosum. Deoxynivalenol is Fusarium mycotoxin and is synthesized by Fusarium graminearum culmorum, especially. Zearalenone also Fusarium mycotoxin and fumonisins also from Fusarium, but other species. Another very important thing that it is well to keep in mind, that the presence of a toxigenic fungi doesn't imply the presence of a mycotoxin. That means that even if we have there a fungi, we have a fungi, we have the bulletin from determination of microorganisms, that doesn't mean that we have necessary mycotoxin there, because some additional conditions are required for mycotoxin synthesis. And in reverse, the absence of any visible sign of fungi doesn't mean that our product is free of mycotoxins. Why? Because during the processing the product, maybe the microorganism is dead, it disappears from there, but mycotoxins still remain. That's why usually you have to do both determination, mycotoxigenic fungi and mycotoxins. Briefly, which are the health effects of mycotoxins? They are genotoxic, mutagenic, carcinogenic, embryotoxic, teratogenic, and estrogenic. I told you not all of the 400 mycotoxins have this effect. No, all the most aggressive I pointed you before. Regarding the carcinogenic effect, please take into account that aflatoxins, they belong to class 1 of carcinogenity. That means they are compounds that can cause cancer disease. And ochratoxin A and the fumonizins, they are belonging to group 2B. The fact that other mycotoxins are not in a group of carcinogenity, it is not so comfortable because maybe they are not studied enough because these studies need long-term and they are rather difficult to perform. I have to tell you that this topic, mycotoxins, is rather new. That group fumonizins were discovered in 80s. So rather new topic.
5 minutos. 5
yes,
minutes.
Yes, quickly. These are the factors affecting the occurrence of toxigenic fungi and mycotoxins. They are biological factors, planting, environmental factors, harvesting, storage, distribution, and processing factors. All of them can affect the presence of toxigenic fungi or mycotoxins. Now, let's see. Regarding aromatic herbs, which are the which is the fungi contamination? As you see, here are some examples of aromatic herb, and here are the identified fungi. As you see here, we have species that I told you before that can be mycotoxigenic. Look here, Aspergillus, Aspergillus niger, versicolor, Rhizopus species. Then for peppermint, Aspergillus flavus, Niger, Aspergillus ocraceus, and so on. So here we can find species that have potential for mycotoxins producing. Oh, sorry. Regarding medicinal plants, here we have a larger contamination. We can say we have a larger contamination. I put here only several examples, but there are many cited examples like this. Mycotoxin contamination. Here I mentioned only the mycotoxins produced by Aspergillus species and Penicillium species. And you see we have aflatoxin. I told you it is class 1 for carcinogenity. We have here in menta aflatoxins, also salvia, also in ginger, aflatoxins, ochratoxin. Ochratoxin A is class 2B, but is nephrotoxic aflatoxin A. There are not so many studies regarding the mycotoxin contamination of aromatic plants and medicinal plants. And I have to mention that a very strong research group is here in Spain, and Santos, Mrs. Santos, is the leader of this group. They have a lot of research done in this field. And mycotoxin contamination with Fusarium species. Look here, we have fumonizins, rather high level. You have to take into account that we have PPBs level here. And for commodities, aflatoxin is regulated in PPB. And deoxynivalenol was present in almost all the samples presented here. Deoxynivalenol is also called vomitoxin. Why? Because it has a special effect on gastrointestinal tract. And very important that deoxynivalenol is immunosuppressive. And things to take home, that aromatic plants and medicinal plants are susceptible to toxigenic fungi contamination. And even the filamentous fungi can be seen, the mycotoxin may be present, and when intended to be used in a food product, it is a good approach to determine the contamination, microbial contamination and mycotoxin contamination of the aromatic herb. Why? Because when put it into a product, there we can have a cumulated effect of mycotoxin from aromatic herb and from other commodities used for obtaining that product. Thank you.